So if you'd like to take your seats, please. We try to reclaim a little bit of time after the really interesting discussions this morning. We've got quite a large panel of panelists, I think is the right term, but we've a large number of them anyway. And we have only 1 hour for this session, so we'll try to make best use of the time that we have. I'm just going to, this session is dedicated to trying to identify areas of collaborative research in which we'll be able to move the field forward. And what we thought we would do is actually the microbiome spreads across many different sectors: food, health, humans, animals, and there are many different actors playing in this space, and they are having different demands and different questions. So we thought we'd like to capture what some of these questions are, identify any commonalities there might be, and then explore with you whether there are some approaches that we could bring into an IMI type program. So I'm just going to ask the panelists to introduce themselves, to tell us about their interest or their organization's interest in the microbiome, and then also highlight for them what they see as the key challenge, and then set that as the basis. It's a panelist discussion, but we need your questions, so please, I hope we can rely on you to ask lots and lots of questions. So with that, I'll keep quiet, and I don't know, which end shall we start? Shall we start at the far end?
Hi, so my name is Vijay Yajnik. Thank you for inviting me. I work for Takeda Pharmaceuticals. I'm a physician scientist by training. I recently joined Takeda as the global lead for clinical microbiome development. Our company is externally focused. We have 2 partnerships, and we're trying to bring consortia-based microbiome therapeutics to patients with Crohn's disease, ulcerative colitis. The biggest challenge we face is first of all, is how do we make consortia? Our partners struggle in developing a pill that be given to a patient. Our second challenge is then how do we measure what we give to the patients? For example, if they swallow 4 pills, 2 pills. So, conventional drug development, we talk about target engagement. What is our target? Is it the microbial community that's broken down in the gut? Is it remodeling? How do we do that? How do we dose them daily, weekly? What should be assessed? So those are some of the challenges that I would like to discuss with you.
Good morning. My name is Christopher Neiman. I'm a chemist by training. I am a member of a company called ProDigest, which basically represents the SMEs, the small and mid-sized enterprises in this panel. As a company, we provide a technology platform that consists of a unique translational in vitro gastrointestinal tract model, which we have developed to represent various species, not only human, but also for animal use. Equally, we have developed applications across different age groups, disease types. And we provide this in combination with microbiology expertise to a variety of partners in the food, feed, and pharma sectors. What we experience in working with those partners is that there's a multitude of challenges that need to be overcome. We have already been discussing some of the scientific challenges, the need for probably a little bit more clarity with regard to the regulatory framework, and I understand the cautious approach that agencies are propagating. But equally, with regard to the appetite from the investor community, a gray area always represents a risk, and risk is something that investors don't appreciate. And I think with SMEs and biotech companies being the drivers of innovation when it comes to translating that science into commercial products, this is a constant struggle to find funding for this very early, very risky stage of product development. Secondly, I think the rigor of science and quality is something that has been mentioned a couple of times. I think it's very important that companies who venture out in this space right from the start think very clearly and actually apply the same type of rigor that you see in other sectors, like in the pharma sectors, in terms of what endpoints do you want to reach, what biomarker strategy are they going to go for. That will not only improve their probability of success, but equally will allow to engage payers, regulatory agencies, will allow to start talking about reimbursement, which very often is something that is forgotten. And I think that end-to-end perspective, even if you're at a very early stage, is very important for partners and companies who want to be active in this space.
My name is Frank Ostrom from the Technical University of Denmark, and here I'm also representing the Global Microbial Identifier. That's a more neutral organization, as well as the EU Project Compare, that is looking preliminarily at sharing data on infectious diseases. And I think that one of the issues that could be a challenge, but also of interest in the future, is really the ability to very, very securely share data and rapidly share data around the world. So basically having an infrastructure where if you have microbiome data, you can upload them, you can decide whom to share the data with, not necessarily go into a contract with a certain company, but simply say, have a look at my data or parts of my data, and we could actually analyze this together, as well as having online access to tools. This is something that we have developed for infectious diseases, where we're really talking about a time issue, because we want to be able to identify whether the two strains are the same, basically within minutes or hours for outbreak detection. So I think that this could also be rolled out into the microbiome area, where companies and especially SMEs could really get access to tools and also comparing their data to other people's data. I think that another area that I would also encourage you to think about, that would be to keep in pre-competitive research. Basically, if you could go out and identify what is a healthy and what is a diseased gut, then the companies could come up with various solutions, but it will allow them to have a common basis of understanding, as well as a common basis of understanding for the regulators later on if they had to approve or disapprove of such a product. I think that could be beneficial. Perhaps also consider that diabetes, obesity is something that's very, very, very long term. So perhaps we could learn something from the short term. You go on traveler's diarrhea. Is this something you could treat? Also something going in and reducing the burden of antimicrobial resistance. I think you can very, very targeted do that. It's also difficult to get people to comply, to do something for 3 years. But I would never change my diet or stop drinking red wine, for instance. No matter what, I guess. That should really be something difficult. But eating a pill for 2 weeks or 2 days, I could do that if it was something targeted. And I think we could learn a lot also of how to influence the microbiome by such targeted studies. And then last of all, as I referred to before, do not forget the animals. There is a market there, and there's also opportunities to do research studies. Mice experiments are very, very expensive. Going out, and buying a pig, at a farm, well, you could do that for 50 euro, and you can just kill it and do with it whatever you want. And that's actually fairly cheap compared to any other experiments.
Good morning, everybody. My name is Dirk Hadrich. I'm working in the Health Research Department of the European Commission, and my job is to promote personalized medicine. And for this, we have a framework, Horizon 2020, under which we have plenty of ongoing projects that are currently funded, still ongoing for some time. And here my job is to analyze the ongoing projects, to see what trends we have in these projects, to see what are the challenges. And then on the basis of that, I try to develop future funding decisions. So I work for the decision making where the Commission should put the money in future for next year, for example. And here I would like to draw your attention to a publication that has just been done 1 week ago on the internet. We published our work program for next year, and we have one topic on the microbiome. We have been working very hard on this topic, and you will see the description. You have to read the topic description on the internet, which is currently only a draft. It can still change. But when you read this, you can see also what we believe, what the challenges in this area are. And the challenges have already been mentioned by earlier speakers today. It's, of course, sharing data. We have collected lots of data. We have to exploit all these data. On the same time, we have to combine it with other data from other parameters. We have to take into account other omics results from other research areas, and we have to integrate all this. We have to share data, and we have to see what is a healthy microbiome. Also, this is a question that we have put in our topic description. And so we come to multidisciplinary work. This is for us what we see in many other projects as well. There is a lot of results from the metagenomics area in the past, but now we are getting multidisciplinary, and we are about to integrate other disciplines, and we want to put all this together to get the full picture for the healthy state or for diseases.
Thank you. Good morning, everybody. My name is Marco Greco. I'm the president of the European Patient Forum. And for the last 10 years, I've been also the chairman of the European Crohn's and Colitis Federation, so representing around 100,000 patients with Crohn's disease and ulcerative colitis in Europe. As it has been already mentioned a couple of times, I'm representing a patient group that is specifically and highly interested in the subject. Our main aim is to start giving the right information to patients, and so I think that my main goal for being here today is actually to ask you not to forget to correctly inform patients since the beginning. Do not make this train leave without us, because otherwise it will be too complicated to run after the train. I'm a Crohn's patient myself since 25 years, and I've been through different ages in the therapeutic approach to this disease. No, at the very beginning, it seemed that the main priority was to clean the bowel. So antibiotics therapy was a very important part of the first approach to a newly diagnosed patient. So clean everything, kill everything you find in the bowel, and then we will see what happens. Now we are learning that it's not probably the best approach, that it's better to maybe to kill something with thinking about what you're killing, or maybe to replace something or to integrate. It's important, as I said, to inform patients, because up to now there is a lot of misinformation about this subject. Patient goes on the internet, do not forget that, and they can find everything and the contrary. And this is a negative thing. The other aspect is that I have to say we are a little bit scared, because over the last 20 years we have been listening to gastroenterologists to debate, and debate is an understatement, about the meaning of healing and mucosal healing. Now, if we have also to understand what is unhealthy microbiome in the bowel, I think that will lead other years of discussion. So we need to better understand what's happening there also, because I believe that the presence of the patient will be essential for improvements in this field, not only because it is our bowel that it is on the table, but basically we can be partner in this evolution, trying to define together the goals and also the benchmarks maybe that we shall use in the next years.
Yeah, my name is Wolf Fischer-Knuppertz. By training, I'm a veterinarian, and you know, in veterinary medicine, the microbiome plays already a stronger role since decades to increase efficacy of production process of cattle and pigs. So now I'm sitting here because I'm the CEO of Biocartis. Biocartis is a company engaged in metabolomics. And fortunately, due to the previous speeches, I don't need to explain the importance of metabolomics in various diseases. It goes into precision medicine. In many questions, it's considered to play a larger role than currently noticed. Its metabolomics is like unnoticed, like the microbiome in the past. It's a kind of extension of clinical chemistry that we are anyhow conducting, like glucose, the most famous one, and this kind of extension, we can measure them now more, and we will get more information in future. The focus of our company is so-called targeted metabolomics. You will hear about profiling and targeted metabolomics. Targeted metabolomics is the only possibility to create metabolomics results and make them comparable from lab to lab and from run to run. So that's why we are in many epidemiological studies established, where you want to repeat the measurement after several years in the same cohort, and you want to compare the results, and you want also to to pool different cohorts measured by the same technology. For that, we are selling kits so that you don't need to send the samples to one central station. You can measure them remotely in your own laboratory. We are part of many consortia in diabetes, of course, metabolomics, metabolic diseases, but also in Alzheimer and in cardiovascular. I would presume many diseases where the microbiome plays a much larger role than currently identified. And I like to talk also in our group to hold the flag up of functional microbiomics, because in the end, I think it's important to understand the interaction between host and microbiome, and exposure like nutrition and drug. And let's say my conviction is that we find that only out when we find metabolites in the blood or in any matrix of the human that gives a hint where it comes from, that we can figure out what the influences are.
Thank you. Good morning, everyone. My name is Antoine Coutan. I'm the Managing Director for Savencia Ingredients and Nutritional Solutions. So from a food industry background, I've worked more than 30 years in food ingredients, mostly for the food industry. And actually, that's what I would like to bring today, is more that insight on food ingredients and food technology than representing Savencia itself. So from a food industry perspective, I think what we need to do today is really to perimeter really well. What are the fields? To me, I see three main fields. One, from the spectrum of the pharma industry, is obviously the study of microbiome and so on into the therapeutic fields, for instance. At the opposite end, we have the food industry, which obviously provides diet and food products and so on, and there's more and more evidence that diet has an impact on microbiome and so on. So the food industry interest here is to see the positive influence of diet on microbiome, on a healthy population. But then the third area, which I think is in the middle and I think is interesting and could overlap between food players and pharmaceutical players, in my view, is patient care. Is basically when there is a patient under treatment, obviously under stress with a microbiome on its own, I think the food industry could bring its know-how and knowledge on the food technology and the way to bring the specialized nutrition to patients and work with the pharma industry in that field. And that's where, for me, I came this morning with this idea of the main challenge is obviously that it is multidisciplinary. We go from nutrition science, which is my background, I would say. But then you have microbiology. The food industry is very well equipped in microbiology. In the ingredient field, you have players like DuPont, like Cargill, like Christensen, people who master the technology. They are involved in probiotics. Then you go into genomics, you go into regulatory, which is a very important competency to have to work on this patient care field, obviously. Socioeconomics, etc., etc. So to me, the key challenge is coming from the food industry background is this middle perimeter, this middle field between food and pharma around the patient into patient care. Thank you.
Okay. So I think it's really interesting that you come from such a wide range of different backgrounds. So now I'm going to ask you quite a difficult question. So Antoine, you've mentioned this idea of collaboration in patient care. So what are those things or those aspects that you would be looking the pharmaceutical industry to bring into that space, and what would your industry bring into that space?
Well, the pharma industry comes with knowing the patient in its condition, its disease, and the way it's being approached with a certain therapeutic approach. And the food industry could come with trying to specialize the nutrition, the food and nutrition intake of the patient. And in the middle, obviously, we have a subject which is the microbiome and the microbiota, because we all know, cancer patients, we all know patients that are under treatment, and we know that very often, there is an issue of the treatment on the microbiota, for instance. Not even talking about, I think we mentioned antibiotic before. Yes, of course, the resilience of the flora with antibiotic treatment and the resilience of certain microbiome profile is a very important subject for that. So I think definitely the food industry is under-equipped, in my opinion, in really the clinical studies that go with taking care of the patient, the analysis of the efficacy of the treatment and so on. So that's where the pharma industry has a strong point. But the food industry, on the other hand, has technologies for the bacteria production at a very, very large scale. And it's today actually being, Savencia is a dairy company, obviously to make cheese, yogurt and so on, you have to make microorganisms. So that's the background of a big part of the food industry is microorganism mastering, if you wish. So these fields can merge, and I'm not even mentioning digital. I was based in California until this year, and that's why I mentioned, you put your TV on, you see probiotics everywhere. But I think the digital companies also should play a role. You saw Amazon just bought Whole Foods. I mean, digital companies are going into taking care of our diet today. So I think all this is merging. It's a great time to invest in that. I think I'm very thankful for the IMI to actually broaden the spectrum of the industries to participate. I think it's a very nice vision. And now we have to go on practicity, defining the fields, the perimeter, and who does what.
Do the easy bit.
Yeah.
So maybe if we go to Vijay, so if you pardon the pun, is there the appetite within pharmaceutical industry to move into this patient space, as Antoine has described?
Yeah, I think in all companies, and Takeda included, we have very patient-centric forums. We start with them. At the end of the day, it really affects. And as a clinician, I remember one of my Crohn's patients, started the patient on Remicade, and she didn't respond. And then I switched her to Humira, and she said, "Am I a guinea pig?" And I said, "Well, no, the drugs are at best 40% efficacious." And so sometimes it's the patient comes in but so worried about the side effects, and when you give them the medication, I'm worried about efficacy and safety, obviously, but the patient's worried so much about safety that when they accept the drug and it doesn't work, they feel very defeated. So we need to provide, as we said, personalized medicine, highly efficacious, highly safe drugs. That's really the future.
So if just a more general question for all the panelists. If you are looking at, so we've heard about nutrition and the pharma industry and what you're looking from them. If I were to ask the other members of the panel, when you look at other industries, are there components of their work or of their data that you think your industry would benefit from? And then maybe we could deal with that first, and then we can talk a little bit about how we could access that. So I don't know, maybe Vijay, if you want to start again, just.
So when Professor Ehrlich was talking, I talked about functional metagenomics. We talked about functional transcriptomics. If I could collaborate with somebody that was really good in stool metabolomics, my program would move forward. So unfortunately, when we got into the microbiome space, we thought Clostridium difficile colitis is the lowest hanging fruit, it should be easily solved. But we see failure after failure after failure because we don't have all the tools. We don't know who truly has the disease, who's just a carrier for the toxin, and who has that bile metabolism that's really susceptible. So I think coming together as a group here, I think would be highly beneficial.
So if we just come back.
I think we also have to accept that, the microbiome is attracting huge amounts of interest. It's a hype, let's be honest. there are huge technical developments ongoing that have led to the point where we are right now, which has been mostly descriptive. I think we're now at the flipping point where we start to build better understanding on the mechanistic end of things. And I think as we dig deeper into the host microbiome nexus, and as we engage in more clinical trials, and I'm just focusing on the pharmaceutical setting, we'll also see attrition. And I think this is a given in any potentially transformational area of research and development. And so we'll see ups and downs, we see highs and lows. I think in the end, we will reach a steady state where we will be able to tap into the full potential of the microbiome. I think for sure it's a treasure trove for minimalistically new targets, and how we approach those targets using microbial-based products or conventional modalities that I think is open. For sure there's huge potential in terms of diagnostics, but I think it will be some time before we reach that real value creation point. I think what is required, I think data access has been mentioned already. I think data ownership, not just in the context of microbiome, but in many other aspects, is a huge issue. I think certainly for smaller companies who don't have the means to conduct large-scale clinical trials, having access to those data sets and to understand from a translational perspective how the microbiome is interacting is hugely important. I think equally understanding how the healthcare landscape is going to evolve is also important. The patient pathway today will not be the patient pathway of tomorrow. We are moving away from therapeutic intervention. We are moving much more towards prevention, interception, and that will require a very different economical landscape as well. And I think anticipating how that economical landscape will look like, how that will translate into regulatory guidance, what the value proposition will look like, will be very, very important. Because we are working not for products of tomorrow, we're working for products that probably will reach a patient in 3 to 5 years from now.
So, well, I think I'll just pick up on the data sharing, and because I think that's still one of the most important things. And equally well, it's important to share when something has failed, because it's really a waste of time if someone else goes down, to do the same kind of studies. And if you just look at scientific publications, we normally have a tendency to publish, positive results, whereas all the failures can be quite difficult to publish and do not get the same visibility. I think also access to data, raw data, to do meta-analysis, but also perhaps access to samples where you can do reanalysis. There's a lot of biobanking that has been going on for a very, very long time. And you're actually stuck with a lot of biobanks, we ourselves are, where we do not even know if we're allowed to do anything with these samples, but we do not dare to throw them out. So I think having access to patients, so you can go back and ask, are we allowed to do further studies on this, because it has really been well controlled studies. I also think that we need to accept that there's new EU regulation coming in force next year, the EU data regulation. Basically the right to be forgotten, which means that if you have a genome, you delivered a sample, you actually have the right to have that removed, not only from the raw data, but also from any subsequent analysis that's being done. And I think that we should be aware of how do we actually enable a system where you can go in and say, well, you have to prove that you have deleted my data, and you also have to be able to prove that you have deleted from any subsequent analysis you have done for the last 10 years. So that's going to be a challenge for the future. But you just might, you just need to embrace that, because it is going to happen. So e-privacy is very, very important.
What we have seen in the past, there was always the focus on one discipline only. So, for example, we have put a lot of money in metagenomic sequencing, and then, of course, there were some great results in this area and new scientific findings. At the same time, there were other projects on metabolomics or on collecting nutritional data, but all this went separately. And now the next step should be to use all these data. We collected a lot of data, we invested a lot in all of these data. Now we have to make sense of all these data. We have to exploit them, we have to combine them, because the diseases are very complex, and we have to take into account parameters from different areas, and we have to put all this together to get, in the end, a really useful tool that the patients can use to improve their health or to prevent diseases.
Well, I'm not representing an industry, but
No, but
say.
maybe.
But I would like to add a few comments
Of
on
course,
what has
yeah.
been said. I think there are three aspects that we have to underline. The first one is cooperation, and is actually, I mean, probably we have to copy a bit the subject of what we are studying. And I know, I mean, microbiome is a complex environment in which different microbes cooperates together, no? And I think that this message of cooperation should be the lead for us. Two things that have been mentioned that were interesting. The first one is about safety, and that is indeed a priority for patients. We have made huge campaigns about that. Informing patient means that the patient is more keen to understand what's happening, but also to understand the risks that he is taking. And to be honest, according to our experience in the patients groups, also on a local level, where it is easier to get this kind of experience, we notice that it's much more easy for patients to take something about microbiome than, for example, to access a biological therapist that is much more scaring. This despite the fact that, for example, the actual product on the market can have very different outcomes on patients with IBD, you know, and probably is once again the demonstration that we're talking about IBD, so we're talking about two diseases, but probably we are talking about a family of diseases that are so different from one patient to the other. So for sure, that's something we have to consider, and I believe it's something that will make patients more keen to take part in trials on that or in cooperative initiative about that. The second thing was about data. APF was in first line in the development of the new legislation, in being consulted about that. But I would like to stress one point, and it is that it's once again all about transparency. Once the patient will know where the data will go and what kind of benefit they can get personally and as a community from the usage of their data, then the risk of the patients to ask to cancel the data will decrease, in my opinion. With this said, we have also to consider that there is a percentage, an important percentage of the population that is not even considering the issue. I mean, if you consider how many have bought a DNA test with some private company, not even thinking that the company will resell the results. So what we're really talking about. I mean, it's important to have a regulation of the data. It's important to create a framework, a clear framework of rules that can somehow give clearer rule of engagement between patient and industry in this kind of cooperation.
Yeah, we are not an active player in that game, but let's say interested observer. And first of all, I appreciate that we are now coming from or seeing a paradigm shift in medicine, which is from genotype to more phenotype. Even oncology congresses are now more and more talking about phenotype. And of course, one part of the phenotype is the microbiome, which is very individual. The challenge to us is to find homogeneous group like responders, non-responders. And the whole thing is very complex. I'm very much driven by the publication that I see done with our technology, let's say every week a new one. And the challenge is on the one hand side to build a kind of bottom-up model, which is comprised of the microbiome, the bile acids, the steroid hormones, and the metabolites, to have a kind of mechanistic understanding. But I see also very valuable publications now, obese people, even inherited obese people, under a certain diet, the weight goes down, the glucose goes down severely. So, and that's when I see such publications, then I think we should not wait another 3 years before we are trying to bring these very promising results already to a kind of routine. So to our group, it's a kind of challenge that we don't move now a couple of years forward, then we are looking to the results, and then we talk another 2 years about implementation. So I think it should be a kind of approach back and forth, and that's what I would like to see in that group. And of course, that requires a kind of close collaboration and a good design of the projects. And I would also say to continuously incorporate the publication that we see every day new, and to challenge what we are doing by such publications that we don't create redundancies.
So, Antoine, before I move to the floor, is there anything you'd like to add in addition?
No, but I really agree with everything that has been said. I think maybe one thing to add from the food perspective, I was very interested into to listen about the regulation, about the risk factors that Professor Sands mentioned this morning. I think that's where also the food industry could focus on the population who carry a risk factor through their diet and through a better study of the microbiome related to the risk factor. And that's maybe one exception of the rule. For me, the main area of interest is still into patient care, because obviously there are work being done on the impact of diet on the microbiome already outside of, I would say, the pure pharma sector. So, but then about that, of course, is all the issues which are multidisciplinary, including the digital aspects, which I think is very important there. And the e-confidentiality, of course, is important too. So.
Great, thank you.
Just make one quick comment on the safety piece, and also dovetails with Nino and Yolanda's talk. I think we're pretty confident the short-term safety is very good in microbiome, but we just don't know what happens long-term. We don't know what would happen to a transplanted patient 10 years. Would they now develop cardiovascular disease that they were not? So that's just as a general point. I don't want to debate it anymore, but that's just important to bring up.
Okay, so Disco, you had a point.
Yeah, I really enjoyed the panel's multiple perspective on the microbiome. Can you hear me?
No.
Yes. Okay, I can scream.
No.
I enjoyed your presentation.
We can hear you now.
And the issues that you raise are, so topical. I mean, the studies on the microbiome we carried out are quite costly, and Dirk had pointed that very clearly. And the need for crossing these things is obvious. But we do have some limitations. Many early studies haven't been done to the standards that we would need. So it's difficult to cross. And we should be aware of the confounding effects of different sample handling collection and all that sort of things. Standardization. Data sharing. We clearly saw in International Human Microbiome Consortium data sharing as far as omics data are concerned. These metagenomics data are deposited publicly available, the data generated across public studies, but we haven't been able to deal with the issue of clinical data, which are simply not shared. We have defined something like all of the 6 fields that are shared, like age, BMI, that sort of thing. But by and large, clinical data are not shared, and I understand issues about that. And of course, the field has reached a maturity where we want to integrate different data types. And we have learned throughout these large projects to handle millions of variables, so it's not difficult for us to add hundreds or thousands of clinical, nutritional, on 10,000, if that could be reached, of metabolomic and proteomic data, but they have to be generated on samples that are comparable, where things are shared. So to me, the issue is what would your suggestions be to go into direction of standardization, clinical data sharing, and generating samples where you could do the integration. The tools are largely available already. We need to improve them, but that's where we stand.
So thank you. So who in the panel would like to kick off with that? Because I think this is very much in the spirit of our discussion today and what IMI has the potential to do. But Vijay.
So let me take that. So typically in clinical trials, we do not collect stool, and that's one of the biggest headaches. We have so many trials, but we don't have any stool in the trials. So the first thing, when I took over the program, we did 6 different stool collections, 6 DNA isolations, and then standardized it with the same Curtis Huttenhauer lab and say, just tell me if there's any variations in storage conditions. So now we at least have the pipeline optimized. The next step is to find a CMC grade drug that I can give it to a set of patients and then start studying. So we're getting there, but we have 0 stool collections. We have serum banked and all these things that you just said we're scared to throw away. We have tons of them, but we don't know, we just can't feed the microbiome pipeline right now because we don't have the stool.
Do you think this is a change that could be easily adapted into the way in which pharmaceutical companies do their clinical trials?
Absolutely. These kits are very easy. Patients can take them at home. You don't have to come to the clinic. They can be easily shipped. All that stability has been worked out. And I think it's relatively straightforward. They're not expensive. And once you have the DNA, RNA, you have it permanently. It's a very tiny tube, so refrigeration costs are also very low.
Okay. Would anybody else?
Well, I completely agree regarding the standardization, and I also noticed the latest study you had, with identifying it was the QIAGEN kits that was the best, at least best reproducible one. We found exactly the same for other sample types, so that was certainly those kind of studies is a major way forward. I think that in terms of IT solution, I think that it's not only a question of whether you're able to handle these data. We can also handle that, at our side. You can handle it many places, but it's also a question of can you really handle it in a way where you can document, complete security and transparency across that. So basically, if you log onto a computer, you should be able to document that you cannot download the data. And you should be able to go in and do analysis where you compare microbiomes, where you do some kind of a PCOA plot, whatever, without perhaps even having access to the raw data, because they might not be feasible. And at least I know that in Denmark we're doing that, and also in the COMPARE project we're doing that for infectious diseases, but then we're also doing it from the Genome Denmark project, where we are also collecting, is of course blood samples, tumor samples, also fecal samples, and have this complete, security. You can also make embassy clouds, so even countries that would not, allow transfer of data from one country to another can, get an embassy where the data are provingly deleted afterwards. So there are some solutions on the way. Of course, a lot of this has also been driven by, blockchain technology, like Bitcoin. I guess you heard about Bitcoin, and if they can use it, if people trust it for that, I guess they'll trust it, for these kind of data as well. So if you trust it for money, they'll trust it for their genome, I'm sure.
Okay. So we're beginning to, we've got a couple of questions that have come from the internet. So what is known about genetic predispositions affecting the microbiome?
I'll take that.
Okay.
So all of us are born sterile. So none of us have visible microbiome when we are born. In a very predictable way, we actually assemble the microbial community. There's slight variations when you come through the vagina or through a C-section, but everybody ends up catching up. But if you have, say, predisposition to type 1 diabetes, then you start seeing dysbiosis. A very famous paper was published 2 years ago showing that dysbiosis comes before, means the microbial community breaks down before the antibodies to islet cells, and then comes diabetes. So there's a 3-step process. So previously we never were sure whether it's the chicken or the egg, but it's very clear from more modern studies that patients that have disease susceptibility end up breaking their microbiome and result in dysbiosis.
And then the follow-on question here is, did any study reveal that good microbes represent a reservoir for antimicrobial resistance that can be, I think that should be inherited or inhibited, I'm not sure, inhibited by bad microbes?
Well, I think I'll try to answer that, and the answer is absolutely it's yes. I think an example is CTXM15 that comes from a cleaver. I think Patrice Norman showed that some years ago, and CTXM15 is a gene that's at least has been spreading worldwide, causing a lot of the problems with broad spreads on cephalosporin resistance globally. So that's one example of something coming from at least something that's not normally causing infections. But you could also have another example that is like the bacilli, and especially Bacillus cephaloformis and other that are being used, as pro and prebiotics. And most of them all then contain the D gene. But as so far as I know, it's not been shown that gene has transferred into a strain that is causing infection. But it can cause if it's transferred. So we're actually using that as pre and probiotic and have been doing for a long time. And then, of course, there's the entire microbiome. You can find that in many different species. But if you look at from the origin, yes, certainly, there's examples of that. But this is a very large area of research. We need to figure out what is the association with what we do, including the resistome, but there's certainly also ongoing studies looking into this, trying to quantify what is the resistome, both the microbiome, but also what can potentially in the future be a resistome. And I know that Dusko's group has been working on that as well. We're also using Etienne Roupe's database for these functionally characterized genes. But at least when we look at it, that part is actually not associated to drug use. This is in animals, though. We look at farms where you have used drugs, then this functional resistome is actually not selected by drug use compared to the one that we already have identified as a microbiome. But I guess time will tell whether that's going to be true also for the future.
So, are there any other questions from the audience? Oh, I think there's one way at the back. I'm afraid my eyesight's quite bad, so.
Thank you so much for the interesting discussion. I have one question. What about your opinion to use the phages, especially in this section, and especially when we are talking about some antibiotic resistance? What about your opinion? Thank you so much.
I'm sorry, I missed the first piece of the question. What type of genes?
What about your opinion to use phages?
Oh, phages. Sorry.
Thank you.
It's okay. So, yes?
Well, I think that it has been shown that phages can pick up DNA and be a very, very efficient way of transferring DNA pieces from bacteria to bacteria. So, of course, it's known that a transduction is an efficient way of transferring resistant genes from bacteria to bacteria. So that's, of course, a risk. But I think that especially in Eastern Europe and Russia has been used for decades. You've been using phages also as a therapeutics. And I think that we still need to have more studies showing, is this something that has a long-term impact on changing the gut microbiome, or is this something that's being used more therapeutically, directed at clinical infections? But I certainly would agree that this could be an area where you should obtain more knowledge. And think that even phage therapy was really a big thing in what was in France, but that was of course before the time of antibiotics.
Okay. So
Just one more comment on this is antibiotic. Series Therapeutics just released their results on ulcerative colitis. And one of the incidental things that vancomycin-resistant enterococcus or VRE carriage is a very significant risk factor. So the patients that were given the series spores all were able to clear the VRE status. So that was just very exciting, I think.
okay. So I'd like to return to Marco as a representative of patients. When you look at a future IMI program, you've mentioned some things you'd like to see patients involved, but how would you like to see patients involved in IMI projects going forward? Could you give us a little bit more detail about that?
Well, I think there is not a definitive answer to that, but if I have to choose something now, I would say, first of all, including patient in designing material for informing other patients, because here it's basilar to create a large community of patients that is willing to be involved. Otherwise, there would be difficulties in developing the future of this work. So that means involvement in the trials. The second issue is involving patients for defining some benchmarks. So defining what are the primary goals that we want to achieve. I'm not sure we can. It will be possible to define what it is an healthy microbiome, but for sure we can define together some cornerstones to be used for comparing and for having at least an acceptable quality of life. Now, usually when we don't know how to, when it is fully healthy or not, we start talking about quality of life. That is a good compromise. So I have many other ideas, but I believe if we start from realizing the first two, we are already halfway to the optimal situation.
So you really think that this peace and communication has to be integral to
For
programs
sure,
going forward?
communication that has also to take into deep consideration issues like safety, data, and also the potential. I would be curious maybe to talk maybe on another occasion about the challenges that the new incoming IP regulation on pharma products will determine in this specific area of research.
Okay, that's great, thank you. And then one specific question for you, Dirk. And I mean, IMI is quite a big program. We do big projects. We're not necessarily the go-to place for SMEs, for whoever else are looking for funding, because our projects do tend to be much larger scale. So can you maybe tell the panel and the audience about other funding streams? You've mentioned one topic that's in the future work program for the microbiome, but are there other sources of funding where actually people who want to work on maybe a smaller scale are able to get started and lay the foundations for future work?
Yes, I have been referring to the health research topic that we have published last week, or 1 week, more than 1 week ago. At the same time, we published other topics. And actually, it's quite a long list, and it's very much related to food and also to the microbiome of plants and soil, and also microbiome of fish, aquaculture. So lots of investments are done in these areas because we also believe that this can have an impact in the end on the human microbiome. There was a lot of interaction between these different microbiomes, and we have really a number of topics in this area. And at the same time, we have one coordinative and support action, because the area is getting very complex, and we have the risk that we are losing the overview in this area. So we have also published one topic for 3 million euro. We want to see a coordination and support action. So the purpose of this is not to undertake any research, just to facilitate the exchange of knowledge between the different fields, and also to look at standardization. This has been mentioned earlier. This is a big issue. What we need to produce more standards, and we want to facilitate we want to ensure coherence between the different fields. And here you have to look into the so-called SC2 work program.
Okay, that's great. So time is against us as always. We don't have a lot of time. We've heard from you all about the areas where there's the potential to collaborate. So if we want to make a success of a future collaboration, not thinking about the science, are there any key things we need to overcome You know, are there any key challenges that the nutrition industry would face working with the pharmaceutical industry, or you know, that patients would face getting so closely involved with the pharmaceutical industry or SMEs? We don't have a lot of time, so if any of you maybe have a few words about some of the key bottlenecks you'd really like to see removed, going beyond data analysis, data sharing, things like that. You think you all get on okay?
I think there's two areas that need to be worked on at the beginning, obviously. One is the regulatory pathways moving forward. Another one is IP, obviously, I think
Yes.
is an important one. But otherwise, I see only complementary industries really for patient care. So taking that as a starting point, then the question is to really identify which topics are interesting, what are the collaborative disciplines that need to be, the transversal approach that needs to be there. And maybe to keep it simple is to choose some very well-defined areas of patient care at the beginning. That's what I would say.
I don't know if many of you were able to attend yesterday's session where we had Aled Edwards going on about open access. Is this sort of open access platform, as he envisaged and we see with the Structural Genomics Consortium, is that something that would work in this area, or would it bring more, or would it cause more issues than it would solve?
Just maybe a last remark about IP and exploitation. If we talk about microflora or microorganism, you know, there's the Nagoya Protocol, which Europe has signed. The US has not signed. So then the question is, who owns the flora at the end, and who, when you exploit it, and where does the value go? I mean, is it the patient? Is it the industry? Is it the state? Mm-hmm. So these are things that need to be a little bit clarified, because there's a new approach to exploiting life science, you know, through Nagoya Protocol. So I don't know if it emerged there. So these would be things to overcome and consider, I think.
One of the things is that if you look at the entire microbiome industry, there are 4 FMT trials in ulcerative colitis, and there are a few trials in C. difficile colitis, and a couple in hepatic encephalopathy. But we just don't know anything else about anything else, right? So if IMI had multiple programs, say let's say in celiac and a couple of other diseases, and there was a common data space where people can come and cross-fertilize each other's programs, I think some of those hurdles will come up, because I don't think there's one fundamental dysbiosis that will correct every disease. And going back to the collaboration between nutrition and pharmaceuticals, there must be some nutrients that could be delivered to these commensals that could fertilize them in the gut. That needs to come together. When the pharma industry doesn't know how to make a product like that. And so unless we collaborate with the nutritional industry, we won't be able to overcome that hurdle.
I regret putting you at opposite ends of the panel, because it's clear you want to collaborate.
If I can add to your question, I think from an SME biotech perspective, I think the whole field is currently waiting anxiously for the first non-FMT products to be approved and being made available to patients. I think much of that product development is actually driven to biotech companies, and I think since the last couple of years, close to $850 million have been invested. We've got about 50 equity investments going on there. And I think in order to allow this to happen for the whole of the field, everything that can be done to lower the risk for these companies to remain active in that space is extremely helpful. And of course, at some point in time, you move from pre-competitive into competitive activities. But I think even for biotech companies, being able to tap into some of the pre-competitive information and capabilities that are being made available through public-private partnerships, through IMI initiatives, is extremely important. And equally, to take away to do this systematic de-risking, taking away some of the uncertainties when it comes to regulatory framework will also stimulate the investor community to be much more active. Because right now, you see a limited number of investors, mostly highly specialized, who are active in this space, yet there's much more that can and probably should be done there.
So, if we have no more comments, I think time has beaten us. However, I'd just like to thank you all for taking the time to join us and have this discussion, because what I'm very happy to say is that a lot of the themes that you have touched upon are actually proposed or could be addressed through the 4 themes that have already been considered for topics. And the first one of those is going to be large longitudinal studies with open access to metadata. So, we've heard that access to data is going to be key to moving the field forward. So with that, I'd ask the audience to thank our panelists, and I'd like to thank you as well for taking the time to join us today.